For exam 1, you're going to basically want to go over your notes along with the notes that Dr. Godovan has, because your notes may include additional things about like the function of a process or pathway. And Dr. Godovan uploads kind of the main topics that were discussed in class. So keep good notes. The second thing that I did in preparation for the exam was I went through all of Godovan's notes and then all of his PowerPoints, and anything I didn't really understand, I looked up online or looked up in the book. And sometimes that requires you to look in the index to find. In this review, I'm going to try and cover a lot of the material that was put in the PowerPoints, and then also some of the stuff that was covered in the notes that were written down and uploaded. And then if I'm missing anything, I'm going to look over the outline for exam 1 to try. Alright, starting off, let's go with the 4 fundamental concepts of physiology. You're going to need to be able to define these, and to define them, it's best to understand them and be able to give examples of them. And sometimes your best way to be able to give examples is by understanding. Homeostasis is essentially the body maintaining its physiological variables within its physiological limits. Some of the variables that may fluctuate might be temperature, pH, or osmolarity. Some examples in which these might fluctuate would be extreme heat or extreme cold. And some of the things the body might do to counteract that would be vasodilation of its peripheral blood vessels or sweating in order to try and cool you off. These would all be considered an external stressor, heat. External. An internal stressor, however, might be something like a virus or cancer. The next fundamental concept of physiology is feedback. This is when the output of a controller is either increased or decreased based on the signal sent from a sensor. Most of the body functions on negative feedback. About 95 to 99% of it is actually negative feedback. And there's only a few examples in which positive feedback occurs. The next concept is redundancy. In redundancy, you have more than one mechanism or system that is able to influence or alter a physiological variable. Some examples would be blood sugar or body temperature. 3 really good examples of redundancy are going to be blood sugar or blood glucose levels, which can be influenced by the insulin secretion or growth hormone production, as seen in the figure above. The next good example would be body temperature influenced by extreme heat or extreme cold. If you were to be in extreme heat, your body would compensate by vasodilating the peripheral blood vessels and sweating. These are 2 independent systems that try to cool you off. In extreme cold, they would be vasoconstriction of the blood vessels and your muscles shivering in order to warm you up. Again, independent systems functioning to try and affect the same variable. The last fundamental concept of physiology is going to be integration. The key part about integration is you have 2 or more physiological systems working together to influence one physiological variable or one end result. The best example of this was provided in class, and that was going to be to maintain adequate perfusion of oxygenated blood to all the tissues. In order to have this occur, you basically need your heart, your lungs, and your blood vessels. So it's going to be your cardiovascular system and your respiratory system. They're both working together in order to move the red blood cells throughout your body and to oxygenate the red blood cells. I really didn't like the diagram, but I did memorize it because you do need to know the placement of this stuff and how it functions. But on an AC, the sensor doesn't know it's an error until it gets to the controller. So I just memorized the diagram and went from there because I didn't really like the way it was because it's not 100% correct. But in the body, however, most of the systems, the cells know that it's an error because they have sort of programmed in logic. Therefore, the signal that they send, the chemical signal, is inherently known to be negative because the controller already has the programmed in logic, and so does the sensor because the cells have evolved that way. So I would just focus on memorizing the diagram and then understanding if a system is providing an output chemical and then another cell is monitoring that output chemical, and then if it goes too high, it sends a different output chemical, which then goes back and is read by the original system, and if it decreases the output of the original chemical, then it's negative. And this is the most important about feedback, is if it's negatively affecting the output of the first system, it's negative feedback. If it is positively affecting it, causing more output, it's positive feedback. And it's usually another sensor cell that It's just monitoring the output and then sending out a different chemical, which the controller system basically also reads and provides a different output, which increases for positive, decreases for negative. So I really didn't like the example of the AC controller sensor system diagram, because in AC, the sensor just reads temperature. It doesn't know that there's an error or if there's a set point. It doesn't know those things. It just sends a signal back to the control system. And the control system, however, has the set point along with knows whether or not the signal is an error signal based on the output or if everything's correct. But this diagram is perfect for the way that the body functions, though. So it's a good example, but it's a good analogy, so that way you can get in the right train of thought. If we were to apply this exact concept to growth hormone and blood glucose levels, if you look at the diagram, if we have low blood glucose levels, then the hypothalamus would basically produce and release the growth hormone releasing hormone, which then goes down to the anterior pituitary, and then the anterior pituitary creates human growth hormone and insulin-like growth factors, which speed up the breakdown of the liver glycogen into glucose, which then enters the blood, and then the blood glucose levels rise, which then causes the glucose levels to be read by the hypothalamus. The hypothalamus then, if the glucose levels are too high, produces growth hormone inhibiting hormone, which then goes to the anterior pituitary, which then the anterior pituitary reduces the levels of human growth hormone and insulin-like growth factors, and that slows down the glycogen breakdown in the liver, which then reduces the glucose levels. So both of these systems already have a programmed in logic for the sensor, which goes to the control system. And these systems, they have the controller and the sensor cells integrated into them. The sensor cells know if something's too high or too low. They have a set point programmed into them. For the hypothalamus, it senses the glucose levels, and the sensor cells say we have, for instance, if we have too low of a glucose, we send this signal to the controller cells, and the controller cells say we need to produce GHRH, growth hormone releasing hormone. This is secreted and sensed by the anterior pituitary. Anterior pituitary sensor cells then send a signal back to the controller cells, which say we need to produce human growth hormone and IGF factors, which is basically insulin-like growth factors. These are then sensed by the liver, and the liver then speeds up the breakdown of glycogen to glucose. And then we have increased glucose levels in the blood, which is then again sensed back at the hypothalamus. We now have high glucose levels. We now want to produce GHIH, which is growth hormone inhibiting hormone, which then those are produced and sensed in the anterior pituitary. Anterior pituitary senses this and then says we need to produce less IGF and HGH. Next, you need to know the difference between an endocrine, exocrine, paracrine, and autocrine gland. I like to think of them as two sets of two, just because it's easiest to remember the difference between two sets of two. So let's pull up the diagram and start off with the autocrine cells. Think auto, self, automatic. So they secrete hormones that act on itself, on receptors that are on the same cell. So they secrete local hormones that act on itself. Paracrine, para, which usually means adjacent or beside. So paracrine cells, they emit local hormones or messengers that act on receptors on nearby target cells, which then we work ourselves back to the endocrine system, which these produce messengers or hormones which are secreted into the bloodstream and flow through the bloodstream to target distant cells. So they target receptors on cells that are in other areas of the body far away. Then the exocrine system, these are mainly like the digestive system and sweat glands and salivary. So we don't really deal very much with these systems in in exam 1, but we will definitely start going into those more in detail towards the end of this semester. So just mainly focus on like the endocrine and paracrine and autocrine, but know the difference between each and every one of those. Next topic, the chemistry of hormones, is huge. It's really important for what we build on next. So let me pull this up so that we can start with it. Lipophobic and lipophilic. Lipophobic, we don't like lipids. Phobic, afraid. Lipo, lipid. Lipophilic, philic, like, philic. Lipophobic, we're scared of lipids. We're water soluble. We like water. We're water soluble because we're polar. So proteins are usually polar. Peptides usually polar. Amine derivatives like serotonin, polar. Eicosanoids, polar. All these are water loving. So, which brings me to a little saying I started, which was water peeps and liponauts. Peep is a networking term, so I have an advantage here, but you guys can just remember my water buddies, my water peeps, and my liponauts. And I just thought astronaut when I couldn't remember. What was it? Water peeps, liponaut, astronaut. And that follows with protein, eicosanoids, amines, peptides. And then liponauts, nitric oxide, thyroid, and steroids. The only trick here is you got to remember that the S doesn't go on water peeps. Just remember knots, and then just try and forget the S at the end. And that'll get you the majority of the way here for remembering all of these. And that's a great place. Understanding the chemistry of hormones will inherently allow you to deduce where the receptor location is, whether it's inside the cell or outside the cell. So, if you're polar, that usually means that you're water loving. If you're non-polar, that usually means you don't like water. If you don't like water, you usually like lipids. So it's usually the functional here. So we'll go over a few key terms. Hydrophilic, hydrophobic, lipophilic, lipophobic. Make sure I don't tongue twist this. If you are hydrophilic, you like water. You're water soluble. If you're water soluble, you don't like lipids. It means if you don't like lipids, you're lipophobic. Okay? Now, the other side, if you don't like water, you're hydrophobic, water insoluble. If you're water insoluble, you're probably lipid soluble, which is lipophilic. Okay, lipophilic, like lipids, equals hydrophobic, don't like water. So you're a hormone floating through the bloodstream, and if you're floating through the bloodstream without help, you're probably hydrophilic. Okay, you're water soluble, you like water. You're flowing through the bloodstream, and well, let's say insulin, for instance, because we had a diagram of insulin. Pull it up here. Your insulin, you're polar, you're hydrophilic, you're lipophobic. You cannot get inside the cell. This lipid bilayer, it's a layer of lipids, prevents you from getting inside the cell. You cannot pass, period. You cannot pass. So in order for that cell to exhibit a response, it needs a receptor that's located outside of the cell, because you, as a hormone that's polar, you cannot get past the non-polar layer of lipids. So you bind to a receptor outside of the cell, and that's how you exhibit a cellular response. Okay? Just remember, if you are lipophobic, you can't get inside the cell. And what is lipophobic? We have lipophobic being protein, peptide, amine, eicosanoid. Okay. What about the other hormones, the lipophilic hormones? Well, those guys, they use a helper. This helper is a transport protein. It binds to a transport protein, so that way it can float through the bloodstream, and then it'll unbind and then diffuse into the interstitial fluid, and then from the interstitial fluid, it'll flow and go up to the cell wall, and because it's lipophilic, it'll go right through the cell wall. The layer of lipids will go through the lipids because it's lipid soluble, and into the cytoplasm. And inside of the cytoplasm, sometimes between either the cytoplasm and the nucleus, there'll be a receptor hormone, and then the receptor hormone will bind to the hormone and then form an activated receptor hormone complex. This receptor hormone complex can then alter gene transcription as it flows into the nucleus. And most of the time, once it's activated, there'll be the transporter that'll transport it from the cytoplasm into the nucleus if it's not already. I didn't memorize a link between the mechanism and duration. And it'll help you on the exam, is to associate the chemistry to the speed and duration. that's the best way to think of it. If it's lipophobic, it's scared of lipids, and it binds extracellularly. So that means that it's fast. It just goes up to the cell and binds to the external cell, and it's quick and it has a short duration. So lipophobic is fast and short. Lipophilic, it's a little bit slower. It has to go into the cell. It takes longer to get into the cell. So lipophilic, getting into the cell, is a little bit slower, but it has a longer duration. I guess you could think of it as if you have it floating throughout the entire bloodstream with a helper protein. The lipophilic has a bigger capacity. You've got the whole bloodstream to hold all of the storage of the hormones being held by the helper proteins being held inside of the bloodstream. So, bigger capacity, longer duration. It's the association that'll help you to remember, but I don't think specifically because, insulin flows throughout the entire bloodstream also. So just think of the association. All right, next thing, the three mechanisms of action for any hormone. I don't recall any specific links where it said, let me pull up the diagram real quick. I don't remember any specific association as if there's a change in ability that is specifically because there was a second messenger. I remember them as all being independent, and then specific hormones just were associated with specific mechanisms that they would use. I mean, when I went through the course, I don't remember hearing about anything that said that mechanism 1 directly causes mechanism 2 and 3, or a direct association of one leading to 2. I remembered it and memorized it as one of 3 or one or more of the 3 possible mechanisms will occur when a hormone binds. And those 3 mechanisms are change in permeability, activation of a second messenger, and gene transcription. And we pull out the diagram for these so you can see how they all function together. A second messenger is when you have a ligand has binded to a receptor, and then the receptor has caused a G protein to basically detach and split, and the active part of the G protein is now bound to an adenyl cyclase, and the adenyl cyclase has now caused cyclic AMP to be produced. This is pretty much, cyclic AMP is the second messenger that was produced by the ligand. That is a number 2. Change in permeability. Glucose being transported into the cell by a GLUT4 transporter by insulin. Insulin has changed the permeability to glucose. We'll see if we can pull that diagram back up. There we go. Here we have insulin has bound to the insulin receptor, and then, okay, we'll skip through all this jumble, and then we get to the GLUT4 transporter. The glucose goes to the GLUT4 transporter, and then it brings in the glucose. I believe this is a great example of change in permeability of the cell membrane, a number 1. Then what about a number 3, gene transcription? Well, yes, insulin does cause gene transcription, but it's a big jumble mess, and I'm not good enough to lead you straight to it. So we'll go with, what's a good example? Oh, testosterone. Testosterone is a steroid, and steroids are lipid soluble, so it goes right through the lipid bilayer, and then it binds to a hormone complex, hormone receptor complex, and then the hormone receptor complex goes into the nucleus, and then the nucleus starts transcription starts transcripting genes and creating mRNA, and the mRNA then flow out and then create proteins. So yeah, I don't want to step on anyone's toes due to copyright issues, so I can't post the pictures of the book or whatnot on here, but I went into exam 1 memorizing all of the hormones and the chemical classes for lipid soluble and water soluble. This is table 18.2, and I probably picked up a few points there, but it's extra memorization, memorizing that entire table. But I mean, up to you if you have the spare time. If you don't, probably just memorize kind of what we've gone over, and then a few of these examples, like the ones that we've discussed, like say testosterone, and then under the thyroid area, I kind of looked over in the book the thyroid gland, and I know I picked up another few questions by knowing and reading that section in the book. But if you're short for time, you want to skip that, and I'll try and give you a few tips on what areas to look over also as we. There are 4 types of hormone effects that can occur. You're going to have kinetic, metabolic, morphogenic, and behavioral. Kinetic, think physics 1. Kinetic energy helps things to move is a definition that you want to kind of remember. Kinetic effects helps things to move. The best example would be epinephrine. I remember that as example and association based on epinephrine is an androgenic classification, I believe, and it is used. If you get a shot at epi, you're gonna be pretty darn hyper. I'm pretty sure that's what I remembered, and that's how I associate it. Next is going to be metabolic. Metabolic changes are going to be changes in reaction rates. If you have an increase in metabolism, you're going to process energy faster and more. That's what I focused on an association with, and the example for this I was able to gather based on reading the thyroid section of the book was it affects the thyroid hormones that are being secreted affect the what was it BMR, basal metabolic rate. That's pretty much going to be your overall metabolism, I believe, and well, that's one of the chemicals that are one of the systems that are increased, the BMR, due to thyroid hormones. So it is does have a metabolic component to it. Next is going to be morphogenic. Morphogenic growth, differentiation. Differentiation is a great association because if you really need an example, a tadpole becoming a frog, it's a huge morphogenic change. The thyroid is also has classification under morphogenic effects because the thyroid is partially involved in causing the change from a tadpole to a frog, and it's also involved in central nervous system development in the fetus. And then also, if you were to have hypothyroidism, you could cause serious mental deficiencies and other deficiencies in the body. So it is a factor of morphogenic effects. I cannot remember. More testosterone and estrogen. That also has a morphogenic effect. Think second level sexual characteristics. Those are directly affected by testosterone levels, not specifically testosterone being injected, but the increased levels of testosterone and estrogen that occur with puberty. Those cause primary and secondary sexual characteristics. A few of you were thinking testosterone all of a sudden makes secondary characteristics occur, and while that's true, it's not the going from 0 to some that causes it. It's the fact of going to some to a lot that cause puberty, from my memory. The fourth is going to be behavioral. Behavioral effects, kind of very similar to as it sounds. Those are going to be pretty much nervous system associated, and testosterone is the best example we can provide for this. In class, we provided an example with deer and the testosterone and estrogen level fluctuations that are a huge behavioral nervous system component. We said the testosterone being directly associated to aggressiveness, and we wanted you to also kind of remember that there were cues or triggers that caused this fluctuation in plasma testosterone plasma levels. We wanted you to remember that some of the triggers were temperature, average temperature. Everything started to cool off as winter was approaching, and then the photo period changed. In winter, you have usually a lower number of daylight hours and more nighttime hours. So it changed in the photo period, and also olfactory cues, which is smell. I believe there were components in the urine that were also triggered, components in the female urine. So kind of remember those 3 things. And one of the other things was the cyclic nature of mass breeding. The more time that there's food in the spring, it's a better time for children to be born. So as the photo period and temperature period and olfactory triggers were present, it started a time of, as you would say, mass breeding. And that's basically the explanation of cyclic nature, mass breeding, babies being born at a certain time. And that was due to the triggers all occurring due to external stimuli. And they also wanted you to kind of remember as the part of the year you had low plasma testosterone levels, and the antlers weren't really growing. And this is also the morphogenic component, antler growth, and then the behavioral component, behavior friendly to other bucks. Dr. Goodovin, Robbie, they were buddies, drinking beer and whatnot, having a great time at one part of the year, and other time of the year they just couldn't stand each other. They saw each other, just wanted to ram heads. They were bucks, life of a buck. So later on in the year, that's when the plasma levels were high. Huge antler growth, huge rack, and change in behavior, hugely aggressive. When I saw this slide, I was also reading the book and covering the anterior pituitary cells and their hormones, and it has 5 components to it. You've got your somatotrophs, thiotrophs, gonadotrophs, lactotrophs, and corticotrophs. Somatotrophs, those are the ones that secrete HGH, human growth hormone, and then IGF, which is insulin-like growth factors. The thiotrophs, hence the reason why I covered the thyroid section. Thiotrophs are the thyroid stimulating hormone producing cells. These are the ones that produce pretty much the TSH that increases or stimulates the thyroid. Next is the gonadotrophs. Gonadotrophs, they produce FSH and LH. These are components that are a major factor for the testes and the ovaries. So I also looked over those 2 organs and the functions that occur there. Next was the lactotrophs. Reading this section also recovers the positive feedback of oxytocin, but it's not part of the lactotrophs. It was just a section also went into it. But the lactotrophs secrete prolactin. This is what initiates milk production in the mammary glands. Additionally, breastfeeding has 2 components. You basically have the prolactin being secreted, which causes milk production, and then you also have the action of breastfeeding. That was the other component of the baby actually feeding. That was the other part that caused the ejection of lactose. So that section goes over that. Next was corticotrophs. The corticotrophs, this relates to the adrenal gland. Adrenocorticotropic hormone, which is produced, which then goes to the adrenal gland, and that has, no, that's a different area. That's what stimulates the adrenal cortex to secrete glucocorticoids, such as cortisol. Hence the reason I covered everything in the chemical compounds list or table that I told you earlier. I saw this slide and saw the pathophysiology area and thought it prudent to also study the tables that related to the hormones of the pituitary and then hormones. Let me pull it up real quick. Oh, summary of principal actions of the anterior pituitary. Summary of the principal actions of anterior pituitary hormones. This was basically taking the hormone, target tissue, and the principal action and goes over all of them. And I looked over all of them for all of the 5 cell types that were occurring in the anterior pituitary. This might help you, but I looked over it prior to the exam, and I know I picked up a few points. I know we did this in class, and I'm amazed how well it worked, even with being awake for 40 hours. So it does work. But yeah, I'm not going over how to memorize that. You guys have that. I can't remember whether or not he's going to give you the keywords, but yeah, from the keywords, you need to be able to go from keyword well, not sound, but necessarily, but keyword to story to name, which then sort of in order, so that way you can get to the number. And that'll give you the 12 cranial nerves. And you need to know that for the exam. I've heard we're going to develop a question on here. So, yeah. Yeah, I enjoyed this part because I actually knew the diagram. I went over it, I practiced it, I understood it. It was 3 points, I swear. It's probably the easiest part of the exam, in my opinion. Yeah, so let me see if I can pull up the diagram, and here we go. Know this diagram. Be able to draw the diagram. Practice the diagram. Take an empty sheet of paper, draw the entire diagram. Be able to explain to someone everything that's happening on this diagram without messing up. If you have to pause, that's fine. If you have to draw it, it's fine. But don't screw it up. Practice it until you can do it without screwing up. Alright, starting with the diagram, start at first messenger. The first messenger is a lipophobic messenger, and it can't get inside the cell, so it binds to a receptor. In second messengers, here we have it binding to a G protein coupled receptor. So in a G protein coupled receptor, the G proteins have 3 subunits. It's going to be an alpha, a beta, and a gamma. The alpha will then, when it's activated, get a GTP, and then that'll activate it. But this occurs only when we have a ligand bind to the receptor. So ligand binds to the receptor, which activates the G protein and gets a GTP. And then once it gets the GTP, it splits subunits, and the alpha and GTP subunit goes floating along the cell wall and hits an adenylate cyclase, the AC, and the adenylate cyclase. This is what produces the cyclic AMP. But from my notes, it basically requires ATP conversion to ADP to power the adenylate cyclase, and the adenylate cyclase also is what converts ATP into cyclic AMP. So we don't get any mix-up, let's pull up what this is happening. It's converting ATP, take 2 phosphates off, and creating cyclic AMP. This is a cyclic AMP. This is a ATP. ADP, 2 phosphates. Cyclic AMP, 1 phosphate that's been bound at, I believe the ester, double check me later, and ATP, okay? Pull this back off. That's what's happening in the adenylate cyclase to create cyclic AMP. You do need to know that cyclic AMP is quickly degraded into AMP. This is a fast action. It occurs quickly, and it's influenced by calcium. So we have the first messenger. It binds to the receptor. The receptor triggers the G protein. G protein triggers the adenylate cyclase. The adenylate cyclase triggers cyclic AMP. The cyclic AMP acts and then is degraded quickly into AMP. All this is being, the PDE is influenced by calcium. After hearing all of you guys' questions or reading about the questions and answers, I decided to look up and find out if calcium calmodulin was required for any PDE action. And it was sort of it's a crosstalk between the calcium calmodulin and its effect on the calcium calmodulin kinase, and then also the cyclic AMP and the protein kinase A, PKA. And there's crosstalk between the two. So it's a little more intricate than I was willing to go into. I want to know that calcium influences PDE, but based on what I read, I wasn't able to give you a definite answer on if it's required for PDE to function, because it's the crosstalk between the two kinases, and if you have crosstalk, it's like this. It's a chicken and egg problem, from what I understand from the material I read. And it's not in the book. This is journal articles. Okay, next is, back to the cyclic AMP. The cyclic AMP binds to the kitty cat ears, the regulatory unit of the PKA. The PKA is composed of a regulatory subunit, which is inhibitory. It's an inhibitory action on the catalytic subunit. So that's a big thing to understand. It's inhibiting the catalytic portion. The catalytic is what does the job. The catalytic subunit is what phosphorylates the proteins. The catalytic takes an ATP and the protein, and it moves the phosphate off of the ATP to the protein, and the protein is now phosphorylated. Okay? The signal is terminated. The second messenger cyclic AMP signal pathway, the signal pathway is terminated once the protein is dephosphorylated by the PPP. The PPP stands for the phosphoprotein phosphatase. Okay? And while the protein is activated, that's when it does its effects. And this is also a key component in what we call the, let's see if I can make sure I don't mess this up, tissue specificity. It's late, don't fault me, you want this review or not. Next is amplification. You need to know where there's amplification and where amplification cannot occur. And that's kind of how you want to say it if you do a free response question. Amplification isn't in action, it's an effect. More than one G protein can be activated by the hormone receptor complex. So right here, that's where amplification can result in the first stage. Then G protein can activate more than one adenylate cyclase, so it can produce more than one active cyclase, AC. Then one AC can make more than one cyclic AMP, so there's 3 modes of action. And then one pKa can phosphorylate more than one protein, so you could have multiple proteins get phosphorylated by an activated pKa. Okay, there's 4 areas, and I actually also memorized like how much. If I remember correctly, it's 10 can be phosphorylated, and then another 10, and then 100. So that's what I remembered. The book will be better at it. So 10, 10, 100 made up my 1,000. I think that's correct. 10,000, sorry. Next is the turning off the signal. This occurs inside of the cell by the PDE. This PDE terminates and it degrades the cyclic AMP. Then you have the PPP. Some people were saying the PPE deactivates cyclic AMP in their questions. I don't think that's the case. The second messenger cyclic AMP pathway signal is terminated when the protein is dephosphorylated by the PPP, which is the phosphoprotein phosphatase. That's the end of the signaling pathway. That's what I remember. I don't remember any link that the PPP degrades cyclic AMP, but I remember that I think the PPP also can trigger the activity of the PDE, but don't focus on that, don't associate that, don't remember that for the exam. I don't believe that's tissue specificity is simple yet complex at the same time. It's composed of the receptors in and on the cell and the intracellular proteins contained within the cell that make up the cell, and the proteins that are contained within the cell at a point in time, because they can fluctuate based on what state the cell is in. Also, it must also contain the receptors for that hormone in an accessible location. If you had like a mutation, that would mess things up. But they have to be in an accessible location for the hormone. And then you could express insulin receptors inside the cell, but if they were inside the cell, you couldn't activate them because they're inside the cell, and it wouldn't invoke a cellular response. Convergent and divergent hormone systems, I actually got away with just knowing what those two systems mean rather than like the association with the examples. However, you may want to learn the examples also and memorize them. So let's pull up our notes real quick for the convergent hormone effect. This is when one first messenger produces multiple second messengers which do the same thing. The best example was epinephrine in the liver. We have alpha and beta receptors in the liver, and epinephrine binds to both of them. And in alpha we have calcium, I believe it's influx occurring, and then in the beta receptors this triggers cyclic AMP to be synthesized. And both of these trigger glycogen to glucose, which I believe is glycogenolysis, I believe. Double check me on it. Glycogenolysis is glycogen to glucose, and glucose to pyruvate is glycolysis, and the reverse process is glucogenesis. So that's what my notes say. I'm sad to admit that I have to memorize it every so often. Next is the divergent hormone effect. So divergent hormone effect is when first messengers will create multiple second messengers, and all the second messengers do different things. Keyword divergent, different things, okay? The best example was epinephrine in the salivary glands. The salivary glands will then have two intracellular protein pathways, and the first one that results from the alpha receptors is the influx of calcium, second messenger, and then the amylase, I believe it's amylase enzyme is synthesized, and the amylase, I believe, is crucial in digesting the food, I believe. Double check me. And the beta receptors, when they're activated, create cyclic AMP through their pathway, and then cyclic AMP with the internal proteins, the intracellular proteins. Those ones are designed for fluid secretion. So before we really go into it, I'm going to just recover the acetylcholine receptor subtypes. Acetylcholine receptors, there's two types. There's the nicotinic and muscarinic receptors. They're two different subtypes of the acetylcholine receptors, okay? And then the other receptor types is we have the serotonin type, and it has 7 receptor subtype families. You don't need to really memorize all of those. I would not worry about it. Just memorize serotonin, okay? Serotonin is one classification, okay? And then acetylcholine receptors are another classification. Okay? The nicotinic acetylcholine receptors are used in a lot of channels and a lot of different cellular proteins that are embedded in the cell wall. Okay, and also another thing to remember is that I believe it's in the ligand-gated sodium channels. It requires 2 acetylcholine because there's 2 nicotinic acetylcholine receptors in it, and it requires 2 acetylcholine to bind to the channel in order to open up the channel and allow sodium influx into the cell. Here we have a muscarinic acetylcholine receptor, and what the acetylcholine is binding it to is a G protein coupled receptor. The G protein detaches with its active alpha subsection and can either bind to a potassium channel. Well, it binds to a potassium channel in the muscarinic receptor action of the parasympathetic system, but we'll go into that again later. But a few of the different options that can occur, or we can bind to a potassium channel, or we could bind to a membrane enzyme. And it depends on these proteins that have been inserted into the cell wall or lipid bilayer. It's their composition that decides what occurs, and I believe that's also the basis of tissue specificity that we went over earlier. It's what is in the cell and what type of proteins are in the cell and if they're in the cell. So personally, we got a bunch of diagrams in here, and if you need more diagrams to understand it, you can take a look at these. They're all pretty much saying about the same thing over and over, but we've gone over and beat it with a stick to death, but we'll go over it again. So let's start off with our acetylcholine. Okay, we have a neuron with its dendrites, and don't really focus on the dendrites over there, just focus on the neuron. And then we have the axon right here, and then we have the synaptic terminal, or presynaptic terminal, and that can be referred to as a few different terms. And you have the terminal bulb, or presynaptic terminal, or axon terminal. And between the terminal and the neuron soma, you have the axon hillock, which is where all the action potentials spike down the axon start. Anyway, that's all you need to know that for other courses and other stuff. But yeah, acetylcholine inside the vesicles. This is, again, I'm going to go a little bit more in detail just because you need to know these things later on. The acetylcholine is degraded by acetylcholinesterase in the synaptic cleft. And then when it's degraded, I can't remember the mechanism that moves it back into the terminal, but essentially you have inside of the presynaptic terminal, you have acetic acid and choline, and then you have a choline transporter that transports that into the That's what it is. The cholinergic transport transports it from the synaptic cleft back into the axon terminal, and then you have the mechanism of proteins that make the acetic acid and choline turn into acetylcholine, and then you have an acetylcholine transporter that transports it into the vesicles, and that is powered by the hydrogen gradient inside the vesicles that has hydrogen pumps fueled by ATP. Anyway, that's all overkill. Don't worry about that. But you do need to know that the acetylcholine inside the vesicles, it gets exocytosed. It exits the terminal by a calcium trigger. Influx of calcium at the terminal triggers exocytosis of the vesicles at the terminal, and exocytosis of acetylcholine into the synaptic cleft. In the synaptic cleft, it will then bind to receptors on the target or the dendritic spine. That's what triggers the sodium channel. Okay, acetylcholinesterase. This is the Pac-Man that we've gone over multiple times that chews up the acetylcholine. Acetylcholinesterase is different from, and we're going to actually skip serotonin because it's identical. Okay, it's identical to this. You can look it up on your own. Acetylcholinesterase, this chews up acetylcholine and breaks it down into acetic acid and choline. The serotonin reuptake transporter, however, it doesn't break it down. It just transports it and puts it back into the presynaptic terminal. That's the key difference between the two. Everything else is identical. And then the other thing you got to remember for serotonin is that if you have low serotonin, it causes depression. Just remember that. There's multiple factors, but that is one of the factors that has been documented. So, yeah. Back to what we were discussing. The acetylcholine usually has targets of muscle, and then glands and other neurons. There's a lot of things that work with the nicotinic acetylcholine receptor. And we'll also need to know, we'll go into the cell membrane potentials and other stuff. Oh, yeah, inhibitors. Yeah, that's a big portion. I saw a lot of people that screwed this up, so don't screw it up on the exam. Acetylcholinesterase is ACHE. It's a Pac-Man. The ghost that I said in class, that they kill Pac-Man, don't they? They what do they do? I have no clue what they do, but they do something to Pac-Man and he goes away. Okay, I don't know. I don't play much Pac-Man anymore. It doesn't. Who plays Pac-Man anymore? The ghosts. They chase Pac-Man. They knock Pac-Man out. Okay, they're inhibitors. They're the bug spray. A lot of people screw this up. It's the inhibitors that stop Pac-Man from destroying the little dots, and the dots flood the synaptic cleft, and they keep triggering the receptors, and the receptors don't stop firing because they keep getting triggered. Okay, that's what happens when you have the inhibitors. Inhibitors are used in bug spray and nerve gas, and anyone who says venom for snakes, that is a concoction of multiple enzymes and different types, and it varies based on the snake venom. So don't use snake venom. We have bug spray and nerve gas. And even nerve gas has so many different variations. There's some that are inhibitors, there's some that are different, okay? But just know that for nerve gas. Bug spray is the best example, but use these in the exam because they're provided here. We can't argue with those two comments if you give them as answers. Okay? Fine. There's one more thing we'll go over. It looks like serotonin uses the G protein coupled receptors. Okay, everything else identical, well, except for what we discussed. And SSRIs are the inhibitors, selective serotonin reuptake inhibitors. Okay, cholera toxin. I actually didn't worry too much about that about the exam. I don't even remember a question on it, but I just may have gotten lucky. So, but let's just go with cholera toxin. It binds to a receptor, and then the receptor is a part of a protein. Protein, I have no clue. Anyway, it triggers a G protein. The G protein triggers an adenocyclase, and then adenocyclase triggers cyclic AMP, and the protein kinase is triggered. So all that happens, and then because of the tissue specificity inside of the intestines, it creates, it causes the chloride and sodium channels to open and water to be effluxed out of the small intestines. So you basically have all these channels getting turned on, and they get stuck on. The cholera toxin binds, and it prohibits the G protein from turning off, if I remember, and it just causes massive loss of sodium and chloride and water through your digestive system, and that's the dangerous side of cholera. Diabetes. Amazingly, there was a massive amount of questions over diabetes, even though it's kind of a simple topic, but it's also in depth. So kind of know what's type 1, type 2, well, I don't know any questions over, but it's good to know. Type 1 is when you're born with it, and type 2 is when you develop. Another thing about diabetes is diabetes doesn't kill you. It's what diabetes causes it. Type 1 is usually a significantly reduced capability of insulin production. It's a genetic disorder, usually, while a type 2 is usually referred to as an adult onset diabetes. This can occur in a few different ways, but at the end of the day, it's usually insulin resistance. It's when your receptors have decreased to a number that you need much higher levels of insulin in order to reduce your glucose and inject glucose into your cells. There's also the concept of that diabetes is the inability to transport glucose into your cells. Not necessarily your blood glucose, but the transport of it into your cells. That's what insulin does. This is not worth a huge amount of points, so we're going to go with this quickly. Normally diabetes turns into heart disease and lung infection. Heart disease. Sorry. Okay, ethnic disparities. We're gonna go through that quickly, just because it's not worth a whole bunch of points. I don't think. Maybe I have no clue on yours, but it wasn't worth a lot of mine. So, this was referring to diabetes. Most of the time, diabetes doesn't kill you. It's the heart disease or peripheral vascular disease that occurs with diabetes that kills you. Diabetes. I don't even see it on here. Oh, here we go. 2%. While heart disease caused by diabetes is probably 17%, and the regular heart disease probably 3%. This is kind of pointing towards the ethnic health disparity. A lot of people in the South don't. Well, they're country. You eat country foods. Lots of grease, lots of oil. I mean, we discussed eating fried butter. Fried butter. Just bad eating habits, okay? A lot of the South has bad eating habits, along with people who are poor have definitely bad eating habits. I don't know. I'm not going to go over the arguments of why. There's multiple aspects. Knowledge, understanding, money, amount of food you can buy. Cheap food is usually unhealthy. Stroke belt, again, matches up. Stroke belt. This was gone over in class. Chronic disease, death by race. I thought it was the funniest thing. Okay, if you want to be less prone to a disease, just get richer. There we go. Downregulation of insulin receptors. What does this mean? We just went over this. Or at least touched base on it. What does it mean? Downregulation of the receptors means that you need more insulin if you have less receptors in order to trigger the same response. What are the consequences of downregulation? Why don't you think of a few? I just kind of gave you an outline for what this could lead to, so go with it. This is another thing. This is the test for it, and knowing that, the test, just understand the test, because you're going to have a question over this. I swear it. And also charts and graphs. And I missed a point because the graphs weren't in color, and I misfollowed a line. So don't miss a point here, okay? You can do this. I looked all over. I couldn't find the other slide that I wanted, but maybe it wasn't here. Anyway, okay, so the glucose test. You fast for 8 hours, then you eat some sugar, and then you wait 2 hours later and you take a reading. And then you take, another thing is to give an insulin injection, and you watch and see how much it drops. So you see how much your body does it, and then you take an insulin injection, and another 30 minutes later you take a look at it and you see how much did it drop. I think on average it should drop like something like, I don't know, like 30 units per 30 milligrams per deciliter per 1 unit of insulin. I think that's the number, but don't quote me on it. There's supposed to be a test. Anyway, you have that much of a drop, 30 milligrams per deciliter per 1 unit. That would be a healthy drop in 1 unit of insulin being injected. But in a person who does not function properly, that one unit of insulin might only decrease it 2 milligrams. That would be an obvious person who's developed onset diabetes. So, next, normal. You need to kind of know this, though. This is big. You need to know these levels. Normal would be 140 or lower. Pre-diabetes would be 140 to 200 milligrams. Okay, fine. Technically, 140 to 199 milligrams. If you have a level of 200 or higher, you're indicative of diabetes because your body isn't able to get your blood glucose levels to drop after that 2-hour period of having the sugar dosage. Okay, like this is your body's ability to decrease your sugar after your blood sugar after eating sugar. Okay, if you have an inability to decrease it, you probably have an inability to get it into your cells, and that's kind of the test analogy here. So a few of the things you might want to remember is exercise is a transporter for glucose. There's a exercise-driven glucose transporter, from what I remember, and then also the hypothalamus is what has the osmolarity sensors, which tells you whether you're thirsty or not. Again, back to a lot of people miss this question, and it refers to osmotic diuresis, and understanding how osmotic diuresis functions leads you to the why you pee a lot and are thirsty when you have bad eating habits and you're diabetic. So let's try and draw this out. Sorry, I don't have a fancy little pen thing going here, so I'm going to go with text. Sorry, buddies. If this is our cell wall, and we were able to get 2 glucose in, and well, we ate a lot of sugar. Now we have a whole bunch of sugar outside our cell and only a little bit of sugar inside our cell. So the transporter is not working very well, and we're only able to get, like, say, let's go with, we were only going to get, like, our sugar down to, like, 8 outside, and our inside, we got it up to 4. So we have twice as much outside the cell, because we can't get any more in. What we do is we start to excrete the water. The water goes out, which increases the concentration of glucose to water. And when we do this, we can get the concentration inside the cell equal to outside the cell, which is the definition of osmolarity, is concentration by volume, I believe. So concentration of particles by volume. I have like an awesome stylus, so I can't do like the awesome drawings off the top of my head. So yeah, I just did text. If we had, this is our cell membrane. Guess I'll type that in for you. Our cell membrane, and it's got like only like 1 glucose inside the cell, and well, our transporter, our insulin transporter, isn't functioning properly in this cell because we're an unhealthy diabetic and we don't eat right, and we're type 2 because we haven't eaten right all our life. That means that we just ate like a whole bunch of candy bars, and okay, we'd have more than 10 in our blood stream, but you get the idea. If you have 10 outside the cellular membrane and 1 inside, the body was able to, we were able to get like maybe like 2 more inside there, or we'll just go with a nice, go with a 6, no, 7, okay, there we go. Now we get a few in here. I'm skipping my math on here, I'm just adding a few. We're almost double outside as inside, and because we can't change the osmolarity inside the cell any more than what we've just done, what we are able to do, because we can't transport anymore. So we change the osmolarity equal outside the cell by excreting water. We'll basically have like water go shooting out of the cell to equalize the osmolarity. Water shoots out of the cell to try and equalize the osmolarity, make it equal to outside of the cell. And when this occurs, because we're an unhealthy diabetic, it causes three things. Well, it causes lots of peeing, and you get really thirsty too, even though you're drinking a lot too. It's because your cells are excreting the water that they're holding in to try and equalize the osmolarity equal to the bloodstream or interstitial fluid around them. You need to know if we have that's how it functions. This is what is this is osmotic diuresis, the excretion of water from inside the cells due to lots of sugar in the bloodstream and your inability to move any more sugar. That's osmotic diuresis, peeing a lot due to that. Now, you also kind of need to know what happens if we're finally able to determine or equalize like 5 to 5. What's inside the blood is equal to inside the cell for the sugar. This is what we call osmolarity. Sorry, isoosmolarity. That's when they're equal. Okay. You also need to know a diabetic may have high blood sugar, but you don't have the blood sugar in the cells. The glucose has not gotten into the cells. Okay. Diabetic ketoacidosis. I can't believe I missed this question. Oh, I couldn't believe it. So frustrated. It was just because I didn't realize that it's the fatty cells that are being oxidized. I was like, okay, I know there's cells being oxidized, and I didn't know if fatty cells were the only cells that have oxidative phosphorylation that creates the ketones. That's what it is. It was. There was a question that said carbohydrates and ketones, and that doesn't occur to my knowledge. It has to be fats. Fats create ketones, and a question that subs fats out for something else makes it wrong. Okay. While I knew what diabetic ketoacidosis was, and that 3 compounds are the big compounds that you need to know that are ketones are acetate, D-beta hydroxybutyrate, and acetone. I may be butchering the sounds, but it's like 1 a.m., okay? You can do this, okay? And then also remember that these 3, this was a question I had, what were the 3 compounds that are created in fat oxidation? They are acetate, D-beta hydroxybutyrate, and acetone. Okay, yes, blood vessels and ketoacidosis, lots of ketoacidosis and lots of hydrogen, lots of acid. Those are some very bad things that occur when this happens. Ketoacidosis is when we have a whole bunch of these acetate and then D-beta hydroxybutyrate and acetone, and when these are created, we have excess free hydrogens produced. Lots and lots of free hydrogen, hence the ketoacidosis. Lots of hydrogen being produced, flowing throughout the bloodstream. And whenever we have lots of hydrogen and acidity flowing through the bloodstream, this causes a pathway in the blood vessels to thicken the blood vessel walls. They get thicker, they get stiffer, and which reduces the. Also, a big factor is when they get thicker, it reduces the ability of oxygen perfusion through the blood vessel, or diffusion rather, to be more precise, and then it decreases the ability for waste to diffuse through the blood vessel wall and get it bound to the red blood cells or be excreted into the bloodstream and then picked up in the kidneys and excreted in the kidneys. All those things are reduced when you have a thicker blood vessel. This symptom of poor these are the things that this is the description that has been given to the state that occurs in peripheral vascular disease. You get diabetes, you have diabetes, and you've been eating unhealthy all your life, and all of a sudden you have a stroke, and they ask, well, where did this occur? What was the cause? And half the time they're like, oh, it was somewhere in the blood vessels where there was a a clump of plaque, and then we had a bunch of red blood cells that got stuck, and then they released and they moved from some peripheral vascular capillary. We have absolutely no clue where it was, because all of your capillaries in the peripheral vascular system are clogging up. So they just say, well, it was somewhere in your peripheral extremities where a clot was dislodged and got caught somewhere else. So this is what we describe as peripheral vascular disease. A lot of the times it can cause some pretty heavy damage. Finally, we're starting to get to the anterior pituitary, and this is one of my favorites because I put the most studies into it. One thing to notice is that the hypothalamus to the anterior pituitary uses blood vessels. The advantage of this is it's a short and direct route, which allows a fast and potent delivery system. I would totally look over the book. There was a great section in the book that really went into detail on this. I highly recommend looking over these subsections of the book. I thought it was a great addition to what was discussed. And it's got nice, pretty pictures. So, I guess going through this, one thing to know is that the anterior pituitary all uses have hypothalamic factors, while the posterior pretty much composes of two major compounds being produced, which is oxytocin and antidiuretic. Antidiuretic is what makes you hold on to water, okay? Hold on, antidiuretic. We went over osmotic diuresis, where we excrete water from our cells and then pee a lot, okay? So remember those two things. The anterior pituitary delivery of hypothalamic factors is via an artery. It's a quick and fast delivery. The advantage of being an artery is that it's short and direct route because it is an artery, okay? It's being pushed out to all the other areas quickly. Fast, direct, potent. Posterior pituitary hormones, oxytocin and ADH, antidiuretic hormone, is synthesized in the cell bodies of the neurons in the hypothalamus, which is then released in the hypothalamus and goes through and then to the axon terminals and the posterior pituitary. See, hypothalamus produced, transported to the terminal, released at the terminal into the posterior pituitary gland. There are some certain characteristics of the posterior pituitary that occur with the fact that it's being transported through a duct into the hypothalamus. There are quite a few characteristics that are involved whenever you have the transport through that duct to the posterior pituitary. This basically has a few advantages. These advantages are you usually create enough hormone in the hypothalamus and sent down the duct into the terminal, and enough is made and stored to last between 12 and 14 hours. And the two chemicals that are being synthesized are oxytocin and ADH. So it does have a advantage of having a much higher capacity of chemicals being stored than the anterior pituitary. Just to go back real quick on the diabetes, just because I don't want to miss this concept, because they will ask you this. It will be a question. What is downregulation of insulin receptors, and when does it occur, and how? Like, what causes it to occur? Rather, is a better comment. Downregulation of insulin receptors is when you get less receptors on the cell wall. It starts to decrease because of excess ligands. Like, if you have excess ligands, you don't need as many receptors to have the same cellular response. So the cell will start to it'll start to absorb some of the receptors and remove them, which can be detrimental whenever you actually need glucose injected into your body and you're eating healthy. If you've eaten unhealthy and raised your glucose levels to super high levels all your entire life, and you get older and you try and eat healthy, it's too late. You've lost your receptors. So downregulation of receptors is a loss of receptors. And essentially, how does this occur? Well, excess ligands. Well, what does that mean for sugar? That means eating lots of bad sugar, lots of bad foods. and constantly having a high blood sugar, and or if you're already somewhat diabetic, pre-diabetic, just abusing your insulin and not managing your food intake and only managing your blood sugar levels. Okay, posterior pituitary and anterior pituitary. I really enjoyed this section, but it's getting kind of late, so I'm going to do my best to go over this quickly because this is in the book, and I've covered it earlier, and it's explained in the book great, and you really do well by we'll lightly go over it. Okay, let's see what we have here. I read about the all construction of the arteries and capillaries and whatnot, and I totally had zero questions over it. So don't really worry about that. You do I can't remember if this was a question about the infundibulum or whatnot, but I remember studying it. So yeah, you do need to know anterior, which is front, and posterior, which means back, and be able to flip this back and forth. See how the brain's oriented? Let's see if I can zoom in here. And scroll A. It's in the front portion for anterior pituitary, posterior pituitary in the back. Okay. Any of you that are gonna make jokes about what it looks like, I hope you make F's. Okay. Anterior pituitary is all running off of the hypothalamic factors. Those are going to be, I believe, these. Okay. There's going to be a few other hormones, and I know I went over them earlier, so take a look at them, read in the book section. This is some of the graphs. I read the graph and looked over this, but the graphs that were provided in class, I recreated them and made them look a lot better, because when I got it, it was all every which way and cut and paste, and it's a pain, okay? So, but these do have correct information, it's just harder to understand, so, and it might be some excess info on there, but essentially you just need to know hypothalamus to anterior pituitary, and then LH and FSH, and they go to specific types of cells. For the testes, you have Leydig cells. For the, they have both actually. They have Leydig cells, which creates testosterone, and then you have Sertoli cells, which they perform the spermatogenesis and create sperm, and also create inhibin. But as we discussed in class, you don't want sperm in your blood. So, because they're sectioned off in a different area, you need some other method to sense the amount of spermatogenesis that is occurring, spermatogenesis that's occurring. And that method that the body designed was to create two chemicals, one that will be sectioned off and set in one area, and the other section will be emitted and used as feedback. That one that is used as feedback is what we call inhibin. One sperm created, one inhibin created. One sperm created, one inhibin created. They're usually supposed to be equal synthesis rates, but then lots of things happen to these bodies. Lots of things make them go down. So yeah, but the inhibin, it stays at a ratio equal to the synthesis, not the concentration, but the synthesis. So that way we can regulate our synthesis rates. There's no way for us to know where these guys go, if they stay or go, because they're sectioned off. So yeah, we use inhibin to provide our negative feedback. Testosterone, again, it's a steroid. Uses a transport protein to go through the cell. I believe through the. It's getting pretty late, so I'm gonna stop here at the anterior pituitary. If you want to look over this, you can. The notes are actually pretty good from what was discussed in class. You need to know the pathways that these go through. You need to know that they're negative feedback pathways. You also need to know how they're different in the male and female. Okay, you need to know where the pathways change and what chemicals are different: progesterone, estrogen, and inhibin, and follicle cells, and the corpus luteum cells, and then also the Sertoli and Leydig cells. Okay, you need to. Notes are pretty good. Go through it, memorize these areas, and also look up a little bit in the book. Okay, to get a good base of knowledge, and we'll continue off later on.